that, I'm going to get out of the way and hand the floor to Carrie Pike.
All right. Thank you, Eli. So can everybody see me and hear me now? Am I good? Okay. Well, thanks for the introduction. We're going to page down.
Page down.
It's not working.
Oh, awesome.
Now it's working. Okay, yeah, I can push a button. All right. So the title of this, slightly uninspired, awareness of genetic principles can enlighten silviculture. Here's what I'm going to try to do today. I am going to give you a snapshot into the world of genetics and how my colleagues in genetics, this isn't my research, but how my colleagues in genetics have provided information, research, data to help us understand how silvicultural practices impact the ecology and in this case more of the evolution of forests in Minnesota. So I had to limit myself. If you love what you've heard, please let us know. There's more where this came from. So this is just a snapshot. I'm trying to cover a lot of different species. And in this case, the principles I'm going to focus on are gene flow, and I will define these a little bit later, inbreeding depression, and we're going to talk about genetic diversity and what to expect about silviculture. We're going to talk about artificial versus natural regeneration, and by that I mean tree planting versus natural regeneration. We're going to talk about thinning, and then we're going to talk about more severe types of harvesting systems where diameter limit cuts versus selection type of harvests. All right, there's a lot of definitions. If there was going to be an exam after this webinar, this would be your study sheet. What I will do is go through each of these. As we're moving along, I'm going to use a lot of pictures to illustrate these concepts. So we're going to start with gene flow. It's a concept in basic biology, but what it means is how genetic material moves across the landscape. There are a lot of factors that affect how genes can move. And in this particular slide, I'm talking about seed. And in conifer seed, I don't know if you can see my little arrow here, they have these little wings, and those wings give it flight. And their seeds are small and lightweight, and they can fly some distances, thereby moving the genetic material across the landscape. This is affected, so the gene flow of seed is also affected not just by the seed, but by the cones themselves. In red pine, for example, those green, ripe cones will dry, but they don't like pop open all the way. They'll open kind of slowly and the seed kind of dribbles out. In jack pine, I have a picture here that shows a serotinous cone and a non-serotinous cone. With a serotinous cone, the cones are closed and the seed doesn't fall out until it's heated and then it opens. And in the non-serotinous, the open cone, the seeds can just fall out at any time. And so the type of cone can affect how the genes can flow. But with broadleaf trees or angiosperms, the seed are not little winged things that can fly around. They're heavy. There's a black walnut here on the left that's going to fall and not move very far, and same with the acorns on the right. So the gene flow in our hardwoods is going to be quite different from that in conifers. So who's responsible for a lot of the gene flow, at least as far as the seed movement is concerned? Our squirrel friends. They are responsible for moving a lot of genetic material of hardwoods across the landscape. And depending on the kind of squirrel you have, if you have a lot of red squirrels around, they have a habit of being something referred to as larder hoarders. This was a new word for me. They disperse large quantities of acorns to few sites, versus our gray squirrels, which are more like scatter hoarders. They'll disperse small quantities of acorns to many sites. So squirrel activity, the species and the squirrels themselves, can have a lot of influence on how the genes are dispersed across the landscape. More commonly in conifers, though, we think about genes that flow by pollen, and so this is the male part. These are male cones on the bottom left of your screen, and you can see they're small and lightweight, and the wind can blow them, and dispersal of pollen can occur over many, many miles. So a lot of the gene flow in trees occurs by wind, both in hardwoods, in conifers and in hardwoods. Insects can help out our hardwoods as well as far as pollination goes. They're not dependent on insects, so they can do both wind and animal dispersal. So there's another term that we may take for granted, a population. And a population is a group of individuals that inhabit an area, and we assume that they're intermating, and that assumption isn't always realized. A group of populations, so if you think of a population maybe as the red pine trees that are outside cloquet here, there may be another population down the road and another behind. We think of like a kind of a stand of trees as a population. These populations might be disjunct, where they're isolated and there's no gene sharing occurring at all, or they might be contiguous, where the genes are shared among the populations. So I'm going to show you an illustration, because I want you to have this in your mind as you're thinking about trees. This is, and not in this particular slide, this is something called disjunct populations. So with plants that tend to be small in stature, maybe are insect dispersed primarily, or they self-pollinate, such as some of our prairie plants and our agricultural plants, they have very limited gene flow. So in this illustration, this is the Crimea River, sorry. There are two different stands, and each of these dots represents a plant. The colors represent relationships. So all these blue plants are related. Maybe they're self-fertile, maybe they just have really heavy seeds that aren't able to cross the river, and maybe this one here actually was able to get some pollen, or some seed did cross this barrier. And here on the other side of the river, this red genotype, it's the same species, but it's a different genotype because it's been isolated and under different conditions. So in this case, we would say this plant, and this occurs more in our prairie plants, you would say this is very structured populations. They're disjunct. They're not doing a lot of there's not a lot of gene sharing occurring. So in essence, what we notice is that there's very large differences between the sites, but within the site, all of these are related. So we have small differences within the sites. But for foresters, get that image out of your head. This is more typically what we see, are these contiguous populations. Trees are tall. That means they can pick up wind currents. Their pollen is light. Their seed, in the case of conifers, is light, can travel long distances. So there's tons of gene flow. It doesn't matter that the Crimea River is in between them. They're sharing material. And it's evident that each of these dots, again, represents a tree in this case. You can see that there's all kinds of genotypes mixed in here. And similarly, on the other stand B, you get this mix of genotypes. The barrier isn't a barrier. It's not a genetic barrier. They can cross it quite well. As a result, you get very large differences from tree to tree. So all your variation is within your site. But if you compared site A to site B, there's no difference at all. The stands genetically are quite closely related. So that's more common for our trees. Back to a little bit of intro biology. Genes and alleles. Genes are the blueprint of all life. We define them as areas basically along the chromosomes or the DNA. And so if this is your chromosome, there's DNA all wrapped up in it, and we consider the gene to be some section of it. This illustration on the right is showing what we mean by allele. So if you take one location on the DNA, you can see there's your diploid organisms. There are two areas where there's a gene. We call this one gene locus, and there's two different alleles at this. And here they're using colors red and blue to illustrate that these are two different alleles at one locus. In reality, nature isn't full of bright colors for us. We have to interpret these gels. And so when we're identifying different alleles, we use these gels in black and white to identify different types of alleles. But for us, we're going to stick with the color theme. So what I'm going to do here is to talk about genetic diversity. It's really, the other term that I'm going to use interchangeably is allelic richness. And what that means is the number of different types of alleles, that's the richness part, that you might have. And so I'm going to illustrate, this illustration here is going to come up periodically. This is what we end up doing when we're trying to assess genetic diversity. We can't interpret the entire genome. Tree genomes especially are quite large. Instead, what we do is we take a subsample of DNA. And we do that, there are lots of resources available to make sure everybody's looking at the same set of them. We use something called molecular markers to identify them. So you get your DNA sample. what this genetic diversity is allowing you to do, what the allelic richness is telling you, it's a metric of, think of biodiversity. We often think of biodiversity as the number of species in an area, but in this case we're looking at the diversity on the DNA itself. So think of it in that way. So again, we select pieces of DNA and we count the number of different alleles or gene forms. So the math is pretty simple. The hard part is actually getting the data itself. And then we can assess the genetic diversity, both of the individual or of an entire population. So for this little individual that I just drew up, there are 6 different loci. There are 12 possible alleles. In my case, I've got some repeating alleles. I have red, yellow, green, blue, and purple. So there are 5 different alleles in my example across 6 different loci. So you can imagine you can also do this and you can study the population. So what I've done here is created a little population. We've got 4 different trees. And what you do is you compare the alleles that go across each locus. So here is locus 1. There are 4 alleles. There's red, yellow, green, and purple. The second one, there's 4 alleles. This one has 5 alleles. This locus 4 has 6. It's got an orange one. This one has 4 alleles, and this one has 5 alleles. And then what you can do is take an average. Say for this population, there's an average of 4.6 alleles per locus. There's an average of 5 per sample. So if you want to answer the question, one way to address this question, what's the allelic richness? It's 4.6. There are other metrics we can use. How many rare alleles are there? Well, there's one that's particularly rare, that orange allele. And its frequency, which is something that's also commonly reported, is out of the 48 locations, there's only 1. So it occurs in 2% of our sample. And there it is right there. So that gives you a handle on, when I say allelic richness, kind of think of these colors and kind of some subsample of DNA. There's another term I'm going to introduce some of you to called heterozygosity. It's often presented as a percent, and that's cool because then it means you can compare heterozygosity among different taxa. You can get heterozygosity for rare cats. You can get heterozygosity for trees. So it's a metric that can be used across them. And what it means, hetero means different, zygo referring to the DNA itself. In practice, it's the percent of your loci on your sample that have different alleles. And I'll show you what that means in a minute. All the other loci are homozygous, where the same allele is present. So let's look at these 2. On the left-hand side, you can see these 2 are different. I have red and yellow. These 2 are different. I have green and red, blue and green, blah, blah. Oh, down here at 6, 1 out of those 6 loci is homozygous, and that means that 5 out of 6 are heterozygous. So I have a percent heterozygosity of 83%. And on this one, it's a little bit different. 3 out of the 6 are heterozygous. So here I've got 2 green ones that line up. I got 2 purple ones, and I got 2 yellow. So 50% of the loci here are in fact heterozygous. The reason heterozygosity is important, it is one of the primary genetic signature, if you will, of inbreeding. And so when we're interested in knowing, do I have an inbred population? Well, you can compare the percent heterozygosity to some baseline population. It can give you an idea of whether or not inbreeding is occurring. So I'm going to use this one again with my 50% to discuss something called inbreeding depression. You've undoubtedly heard of it. The depression piece is when the biological fitness is reduced when mating is occurring among related individuals. So when related individuals are forced to mate, recessive alleles may line up, and that way they make it unmasked. If these recessive alleles are deleterious, then you see some kind of depression in the fitness of the population. So that's what the term means. So when does this happen? Well, it doesn't happen often in trees. We think of it in populations that are isolated, in case prairie plants, that can happen more commonly than in trees. But it's any time that the gene flow is cut off. So you don't have new genes coming in. You're forced to mate with your relatives and your neighbors, and so that gene flow is cut off. Folks who study fisheries biology, for example, may see this in fish populations when dams are formed. So there's your barrier, and then the fish upstream and downstream can no longer meet each other as easily. Okay, let's get into some forestry here. So how is the genetic composition of forested stands affected by these features? So we're going to look at, I will go through, these are my different examples. I'm going to start with natural regeneration in Shelterwood cuts. And I'm using eastern white pine. I know there are problems with growing eastern white pine. This is an example, though, that could be, in some respects, mirrors other species as well. So let's start with this one. Okay, so what is a shelterwood? A shelterwood is a harvesting and regeneration method by which a few vigorous trees that have ample live crown remain, and they remain as a seed source for natural regeneration to come underneath them. So if that's your forest and you take those away. But the question is, what's the optimum density of these residual trees needed to regenerate a vigorous and genetically diverse stand? Is there some number that you need? So there's this neat study that took place at the Menominee Reservation. The author is here, and I'll have a list of references at the end. They looked at two types of shelterwoods. One of them was a high-density shelterwood. So they had between 20 and 40 trees per acre, and this is more of a true shelterwood, where they're providing some shade to the trees beneath it. They also had a low-density shelterwood. This was 6 trees per acre, so this is more of a seed tree harvest than a shelterwood. And then what they did the harvesting, and they took DNA samples from the seedlings that were growing underneath it. So what did they find? Well, as we would expect in our conifers, there's lots and lots of genetic diversity because pollen blows around and the seeds can move. But they did find some inbreeding occurring, which you don't commonly find in pine. Incidentally, these stands were isolated. They weren't surrounded by pine stands. Part of their selection criteria was to not have them be surrounded by other pine stands. So they did find signs of inbreeding depression. So the question is did they find high genetic diversity under this type of a stand or the high density stand? And I'll give you the answer. It was the high density stand. So the genetic diversity of those seedlings growing underneath it, not the parent trees, but the seedlings growing underneath, turns out that it was higher for the higher density shelterwood. So there are several questions. One of them, why? Why would this occur? They concluded that by having more mother trees, there was more genetic diversity. And the reason that is the case is that even though seeds can travel and they have nice little wings on them, most of the natural regeneration in these stands, which were somewhat isolated, was born from the residual trees themselves. So seedlings that were in this high-density shelterwood experienced less inbreeding than those that were in the low-density shelterwood. So the effect of having more genetic material to contribute to the progeny's gene pool counteracted any other benefits of increased light or airflow. You would think some more airflow might allow genes and seeds and pollen to move in more, but in fact that wasn't the case. So I'm going to segue now into the next couple of papers. These next two papers were based on the same type of stands, but they used different molecular techniques. And so this study is also eastern white pine. And they looked at how the genetic diversity of the overstory trees changed after 2 different thinning regimes in old-growth eastern white pine. So a slight shift in what we were just looking at. So they started with, they had 2 uncut EWPs, eastern white pine, old-growth stands in Ontario. The average age of the overstory trees was about 250 years. They had mixed conifer and hardwoods in the understory. So think of this mixed stand with some big trees. What they did was they applied some partial cuts. So they took out some of the old growth and they took out some other merchantable material as well. And in essence what they did was they removed 2/3 of the white pine basal area in one stand and about 70% in the second stand. Very close to each other, so the stands served more as a replication than a treatment itself. And what they did is they extracted DNA from 120 overstory trees in A and 102 in B. And this is from their paper. We redrew it a little bit. And what they have here are some parameters and the pre and the post-harvest information from stand A and B. So you can see the heights. We also converted these to English units. So 85 feet and 89 feet, and after harvesting, the average height increased a little bit here. DBH was about 21 inches, and then 26 or so after harvesting. The basal area went from 258 to 86, 291 to 86. About 11 feet to the nearest neighbor, and then 44 feet to 49 feet after. Hit 132 white pine down to 25, 106 to 23. So what did they find? So this is showing the number of alleles and the percentage of alleles. And what they found, and here's the total, what they retained after harvesting and what they actually lost. So let's look at stand A. So they're looking at, these are the alleles that were unique to stand A. Another term we use for those is that they're private alleles. So there were 12 alleles that were only in stand A. After harvesting, they still had 5, but they lost 7 of them. In stand B, they had 7 private alleles. After harvesting, they had 4 of those alleles left, which means they lost 3 of them. So what they concluded was that harvesting about 75% of the trees resulted in significant losses of allelic richness. The allelic richness dropped 26% in both of the stands. And most of those losses, though, were among alleles that were rare or private. That is, those alleles that either occurred in low frequency, like the orange allele here, or those that were just only common to that particular stand. So what are the implications for eastern white pine? Well, what they suggested is that old growth stands should be maintained on the landscape as reservoirs for these rare alleles. But some of you in forest, you're going, that's all we have is old growth white pine in Minnesota, so what do we do? Well, you know, there's a need to want to balance our stocking of white pine, especially in the northern third of the state where it's not as weedy, so that tomorrow's old growth will foster genetic diversity for the future. So, yes, by losing our old growth, I mean, they're going to blow down eventually. We need to make sure we have the next old growth in line. So, I'm going to segue into another one. We're going to look now not just at, we're going to look now at harvesting that. So that old growth study, they were removing, they weren't doing any real selection. They were cutting some of the old growth, but they weren't imposing a strong selection gradient on it. Now we're going to look at, well, what happens if you're going to really impose a gradient, where you're going to say, I'm either taking the big trees out or the little trees out.
Hey, Carrie.
Yes.
I'm going to interrupt for just a moment. We have not seen any questions coming in, but I want to remind folks that if you do have questions, look for that chat pod. You can see the icon, I think, near the upper right corner of your screen, or else on the green bar on the top of your screen if you have Carrie's slides filling your screen. Go ahead and type the chat, type your question into the chat pod. It would be best if you send it to me, Eli Sagor, but you can also send it to Julie, the host. Either one of us could handle that. Just a reminder, send those in anytime, and as we get questions, we'll convey those to Carrie. Thank you.
Yeah. All right. So what if you're imposing some type of limit on what you are removing from the forest? And so there were a couple studies that looked at this. So let's start with some definition. So we're going to start with the diameter limit harvest. In a diameter limit harvest, you're removing all the trees above a threshold diameter. So this may result in high grading in extreme cases. So you're taking out the big stuff. And in my little cartoon, you're leaving the smaller stuff. In selection harvest, you're truncating it the opposite way. You're removing all the trees with some below some diameter threshold. So what you're doing in here is you're increasing those value of the bigger trees. Maybe you're favoring these bigger trees for a veneer market or something. You want to take out the small stuff so the big stuff will put on more diameter. So there were 2 really cool papers that we can kind of compare and contrast. One of them dealt with black walnut, and their truncation was 30 centimeters, but they did multiple truncations. This was a neat paper because it was a simulation. They genotyped all the trees, and they mimicked what different harvests, the impact of different harvests would be on the genetic makeup. The other study on hemlock, they had a similar truncation level, about 24 centimeters for the diameter limit harvest, but they actually conducted the harvest over 3 3 different periods from 1952 to 1992. So we can see the effects of these harvest types on the genetic makeup. And again, the diameter limit, we're taking out the big stuff. These 2 papers also had selection cuts. And again, they're sort of reverse truncation. So with the black walnut, they harvested them just less than 30 centimeters. Again, that's about 12 inches for those of you in a metric or who prefer English units. With a hemlock, again, it was less than about 9 inches. And in that case, you're favoring the bigger material. What did they find? Let's start with the hemlock. So these are some of the parameters. You can see the number of trees for the control. They had a control. So 35, 31, and 40. They had about 1.5 alleles per locus. But the significant differences were here, the effective number of alleles per locus. And what they found was that the diameter limit cut was actually significantly more alleles per locus, so more genetic diversity than both the selection cut and the control, and the selection cut and control were similar to each other. In terms of heterozygosity, that other term that can also mean genetic diversity, where was it higher? It was higher for the diameter limit cut where you were taking out the bigger material, and statistically similar for the control and the selection cut. Okay. So what they found is residual trees had similar levels. The diameter limit cut had higher levels of genetic diversity, both allelic richness and heterozygosity, than the control. And this was not an expected finding. Why did they find this? Why was the genetic diversity higher when you were taking out the big stuff than the selection cuts? What they concluded was that rare private alleles that were more commonly found in the small diameter class trees. So by taking out the smaller diameter class trees, those rare and private alleles were in fact removed. And these rare alleles might seem like, well, who cares? These are likely alleles that code for poor form or slow growth. But I think in this world, and many of you would recognize that sometimes there's a little fable that talks about the importance sometimes of being the slower grower. These alleles may code for traits that might not seem that important to us for growth, but may have important adaptive qualities in the long run. So they may have value that we're not aware of, so we don't want to discount the finding. Let's look at black walnut, the black walnut study. So they did several simulations. So their treatment intensity, so with this greater than 60 centimeters, that meant they harvest 7% of the trees. Here they harvested 23%. And in the most extreme one, with a 30 centimeter truncation, they harvested 70% of the black walnut in their diameter limit cut. But the allelic richness didn't make any great changes. It does drop off. It went from 17.1 down to 15.4. Here the percent harvested dropped a bit, and the alleles retained also makes a dive. So here you have 99% of all the alleles were retained here. Here, and it's 97, 94. There's a bit of a drop-off here from 94 to 85% of all the alleles in that stand retained. But none of these differences were, in fact, significant. There was also no significant differences when you imposed it the opposite way, so either for diameter limit or the selection value-based harvest scenarios. So what they concluded is that the alleles, all the diversity was in fact evenly distributed across size classes. It wasn't limited to big trees or little trees. But they did end up losing, in the most extreme case, about 15% of the alleles in the most extreme diameter limit treatment. One thing they stated is they think this finding was in part due to high levels of gene flow. The stand history was good where it wasn't isolated. There were a lot of factors that kept the stand genetically pretty diverse. So back to the little colored circles, but what you'll notice in this diagram is I have them on size. So here the colors, and I'm sorry if you're colorblind, the blue ones are all related and the green ones are related, so the like ones are related, but what you can see here is the size. So this is simply my illustration. This is not part of the manuscript, But what you can see, what they concluded in both of these studies is that the alleles were fairly well distributed across the size classes. So if you can see here, for big trees there's red, and there's small trees that are red, and there's big trees that are blue, and small trees that are blue. So if you took out one age class, if you took out all the big ones, you're still leaving genetic material behind. There's not a great loss. So again, to reiterate, the difference between the studies, the hemlock one was cut multiple times, so the effects was more severe than the one-time simulation harvest. And that might be why they found significant differences when the black walnut study could not. But in both of the studies, I think the take-home is that the forest stands were genetically resilient to these harvests. They took it pretty well. What we are likely sacrificing are some rare and private alleles that may be associated with the traits that we're selecting against, and generally they're associated with slow diameter growth. So what do you have to think about then? You're in a stand, you want to know, if I do a diameter limit cut, is it going to have some negative implication on the stand? That you have to use your eyes and you have to understand your site. How successful was prior recruitment? Have you had any problem getting germinants? Did the stand go through some type of bottleneck, a physical bottleneck, something, a road or something large that could have blocked either animals, if it was hardwoods, or seed from coming in? And so think about gene flow. If gene flow is able to occur, you have a better chance of having a successful stand that can naturally regenerate, and it may be more resilient to the type of harvesting that we're discussing here. And if you're at all concerned, if you're not seeing a good response after a cut, you can always add more genetic material. You can plant more trees if you're not getting the response that you need. So I'm going to segue into the next study. Do you have any?
Yeah.
Okay.
Yeah, question related to the last point you just made. Is managing genetic diversity in production stands important if artificial regen is planned after the final harvest?
If you're planning to artificially regenerate, I would say that I don't think you need to worry as much. So I would say no, you don't need to worry as much. If you're relying on natural regeneration, though, entirely, then you need to consider it in your management plan. Okay. So now we're going to look at that question of artificial versus natural regeneration. And so I picked this study. This is a recent study by my colleagues in New Brunswick. This paper came out in 2014. And they looked at the genetic variation of old growth stands, natural regeneration, and plantations. And they had plantations with wild seed sources and improved seed sources. And this particular paper was in white spruce, and they were looking for a genetic footprint. Could you see a genetic footprint depending on the regeneration practice that you use? So for those of you who don't know about improved seed, I have to just put in a little plug about what it is. Genetically improved seed in Minnesota is planted commercially by members of the Minnesota Tree Improvement Cooperative. They're listed in the acknowledgments. Genetically improved does not mean genetically modified. We're just relying on natural variation. We're not inserting genes into this to improve them. Improved means that we increase the expected growth rates by selecting mother trees and constructing seed orchards by the winners through testing. So in Minnesota, we only have improved seed for conifers. To my knowledge, we don't have any for our native broadleaf trees. Where can you buy them? They may be available for purchase from some private nurseries sometimes, and occasionally the DNR state nursery. So where does improved seed come from? It comes from an orchard. This is a picture, an older picture of one of our white spruce grafted orchards. So we may graft the trees, but the seed is open pollinated. So the trees are grafted, and then we collect the seed, and then it's either sold or it's grown into seedlings for planting. Okay. So let's look at the paper. So they sampled old growth natural and sites planted. They had open pollinated seedlings. This was the improved source that were planted at various sites. So what did they find? I'm going to put a table up and I'm going to explain to you what these letters mean. So here are the 4 types of stands: old growth, natural regen, plantation, and improved. So N is the total sample size, the number of trees. AT, this is genetics jargon, is the total number of alleles. So there's 109, 100 here in the improved. The number of alleles per locus, there's a slight decline, but not much. and actually this was the highest. The number of private alleles per locus, there were more private alleles here in the old growth. And then the heterozygosity, which doesn't do a whole lot. But here's the thing. None of these genetic parameters were significant. So now there were some trends, but there were no differences. So what they concluded is that genetic variation was similar among the old growth, the natural region, and the plantations for these stands of white spruce. The genetic base of the planted seeds was broad enough to maintain sufficient genetic diversity. There were bookends. In terms of genetic diversity, the old growth and the tree improvement selections were the two bookends, and you could kind of see that in the allelic diversity. The old growth had more of the private alleles, the improved stuff didn't. So it's kind of bolstering the argument that these rare private alleles are more likely associated with slower growth. So what do they recommend? Well, maintain the broadest genetic base by relying on a variety of regeneration methods. So you don't want to plant improved everywhere, you want to plant some improved. You want to do some natural regen, and again, maintain a reservoir of old growth material. Yes, Eli, is there questions that we can address?
Yeah, should we be collecting more seed from old growth stands to capture rare alleles? Should this seed be bulked with other seed in the seed zone or kept separate?
Good question. I would say it would be worth our time, given climate change, to start bulking, doing some targeted old growth collections from select species. I don't think that's a bad idea. So that should be a separate, I mean, that's a good project to do. Secondly, though, I would say the old growth should go, if it comes from a seed zone, it should be bulked in with a seed zone. It should be kind of part of that mix. If you're getting seed from a seed zone, you expect it to be widely diverse, and that would contribute to that diversity. Anything else?
Nope, that's all I've got.
Okay, I'm going to segue now. This is the last paper, so we're good on time. We're going to talk about hardwoods. I'm going to get out of my comfort zone a little bit here. Hardwoods difference from conifers. They are wind-dispersed pollen, but the animals do a fair bit of dispersal on the seeds. Our favorite critters that we try to keep out of our backyards. So this was tough. I had two papers, and I stuck with the bur oak, and I didn't do the red oak one. This was a really cool study. So this study looked at an abandoned pasture of bur oak. So bur oak that came out, think of, I think this was in Illinois, and they were looking what the genetic value was of the bur oak in these abandoned pastures. So you have a piece of land, it's surrounded by other farms, and the question is what's going on in the stand? Is there any genetic value to it? So this is only one site in Illinois, and what they did is they genotyped all of the trees. They did the adults, they did the saplings and the seedlings, and they studied the relationship between the parents and the successful offspring. And they studied the gene flow that occurred within the stand. So little known thing, we can use, we can do paternal tests on trees to figure out who the dad was. And this is an example of a study where they did that. And so Dow and Ashley are the authors. So let me give you some of the characteristics of the site. And I don't remember how many acres the site was, but the average distance between the adult trees was about 40 feet or 12 meters. The average DBH was about 35 inches or 88 centimeters. They sampled 143 saplings that ranged in diameter from about 1/4 of an inch to about 4.5 inches, and they were less than 20 years old. So what did they find in this isolated stand? So there were 62 adult trees, and out of the 62, 18 of them had served as a seed parent to a sapling, at least one. So about 30% of the adult trees contributed to the stand. 23 of those adult trees served as a pollen donor, so about 1/3. The average distance of the offspring from the maternal tree, so how far did the acorn fall, was 75 to 85 feet. What was the average distance of effective pollination? Quite far. Again, reiterating that pollen is light and can travel. Acorns are not. So the effective pollen distance was close to 200 feet. And 6 of the saplings were immigrants. They didn't share any genetic material maternally or paternally with anybody in the stand. So what does this suggest? what they observed, and again, this is again one of my conceptualized drawings for teaching purposes, and it's not associated with Dow and Ashley 1996. But what they found was that, I'm using my colors and sizes again. So if these big circles represent the big old mother trees out there, they observed that there was actually, the mother trees were pretty distinct. So the maternal genotypes, there's a red, a yellow, a blue, they were pretty genetically evenly distributed. So early recruitment was successful in bringing different mother trees in. They weren't all related. They weren't all red. This hatched one here, by the way, is a stump. What they observed was the stump. They genotyped it, and they found some of its offspring lingering around it. So the squirrels did their jobs in the stand. They were very good at bringing in a lot of different genotypes of oaks to repopulate this abandoned pasture. But they also found that 4 of the adults were seed parents to a large percentage of saplings. And I tried to show this by, if you look across, you'll see there's a lot of yellows and a lot of reds and some greens. And so I tried to illustrate that. So some of the adult trees definitely contribute the most to the stand. But there was a lot of pollen that came in outside of the stand. They also observed that the offspring were clumped around the mother tree. So I tried to illustrate that by showing that there was this clumping occurring. Now, the average distance was, what, 75 feet or so from the mother tree, but there was a distinct clumping that occurring. The offspring weren't randomly scattered. So while the squirrels brought the mother trees in, the offspring that came from those mother trees were pretty well clumped around the mother trees that they came from. So what are the implications? Well, it means that even in isolation, this particular hardwood stand, you can have regeneration that can be pretty genetically diverse if you have good gene flow, and in this case there was. The acorn does not fall far from the tree, but the squirrels did that great job of initial recruitment. So be aware that the stand, now with the trees in place, there's a lot, the genetic material is coming largely from inside of it, but initially it was established from outside material. Pollen is our important source for off-site genetic material, so pollen flow occurs and occurred a lot, and we can thank our squirrels for doing a very good job. I don't know who did it. It was a red squirrel or gray squirrel, but clearly they were the players here that got this established. Okay, so this is my summary slide. I've got a few more minutes, and we should have time for questions. So I'm going to go through, I've got a bunch of bullets here. But I think the lesson is that through this type of research, we can learn a lot about how genes flow across the landscape and how silviculture impacts the evolution of the forest. So what did we find? In general, most of the genetic material originates from trees on or near the stand, even though conifer seed can travel, it doesn't travel that much. There is a lot of pollen flow that comes in on, though, but it's still, a lot of the trees may be sired from the above trees. Shelterwoods, where you have more mother trees, were favorable overseed tree harvests. So again, bolstering that initial bullet. But it's important to know that even when we sacrifice some genetic diversity, gene flow can counteract it, and we've observed this in some of these studies. I do want you to note, though, that inbreeding in trees is it's typically manifests as low seed set. You know, if you're going to see crooked or funny looking trees coming in, they're probably not inbred trees, as you might suspect from folk literature. Instead, it's usually low seed set. So it's the absence of germinants is an indicator of an inbreeding problem. But you do need to pay attention to the vigor of your regeneration. If you've got weak vigor, low germination, you may have a problem with a silvicultural prescription or some forest health thing lurking around. So if you're seeing weak vigor, you're seeing germination, but some weak vigor, there's a silvicultural piece that needs to be addressed. So when in doubt, when you're concerned something isn't working right, you can supplement and feel good about planting trees from your seed zone or improved seed if it's a site where you have good access and would like some better volume growth. Improved seed, and this was just white spruce, is genetically diverse, but it may have a different genetic makeup. So especially compared to the old growth stands, as we saw, it was a bookend genetically with the old growth stands. So improved seed should be part of the prescription. It shouldn't replace all other regeneration mechanisms. So old growth stands may have some special value, harboring rare or private alleles. As the question indicated, we may want to take some efforts to conserve some of that material and be maintained actively as part of the landscape. I think it's important, though, to note that our forests are pretty resilient, at least genetically, to the silvicultural treatments, even those that favor large or small diameter trees. So genetic diversity, at least in our trees where there's a lot of gene flow, was well distributed across size classes. So if you took out all the big trees, you didn't remove all the genes, you didn't extirpate a particular genotype. Remember in my diagrams it was red, orange, and yellow. Those genes are pretty well distributed. This is the case in the northern boreal forest. This is not common in the tropics. In fact, in the tropics, gene flow is not as widespread. It's a very different dynamic. But here in our forests, this appears to be the case. So I would conclude by saying that overall, the silvicultural work we're doing is beneficial. How so? Well, we supplement genetic diversity, and we can help overcome germination barriers by tree planting. We can facilitate natural regeneration by creating disturbances. We can also maximize genetic diversity in a landscape simply by maintaining a balance of age classes. So if you've got 10% of your land in old growth and you've got some proportion in earlier stage, in just doing that, you're very likely maintaining a balance of genetic diversity as well. And so I would encourage you to keep up the good work. This is all the literature I cited, and again, there is so much more where this came from. I had a very hard time picking papers, so I hope you liked the mix that we chose. There's more where this came from. And I do want to acknowledge the members of the Minnesota Tree Improvement Cooperative. Our members are listed here. We are looking to increase membership, particularly by industry, so if there's anybody you think might be of interest, please have them contact me. And my other acknowledgments, Andy David, who's listening in Grand Rapids, and I have Jim Warren here, and Egon in Grand Rapids, Alan Eck, Greg Cuomo, the Cloquet Forestry staff, and especially the SFEC, Eli and Julie. I think that's my last slide, so we're going to just hold it there.
All right. Thank you, Carrie. That was excellent. You covered an awful lot of ground. A couple of thoughts. I think we are going to go ahead. I'll work with Julie to make sure that we send out that list of the literature that you cited. I think you cited a number of studies that were terrific. We can do a screenshot of that, and or somehow, one way or another, we'll include that in the follow-up email. And everybody who's registered for the webinar will receive that email as you do every month. The question that I have here, I only have one right now, maybe a few others will drift in, and this is a follow-up to the question that was asked earlier, Carrie.
Okay.
That question, as a reminder, was should we be collecting more seed from old growth stands to capture those rare alleles, and should this seed be bulked with other seed in the seed zone or kept separate? The follow-up question there was, okay then, how would we evaluate stand performance based on, the collected old growth seed and so on? Was that clear, or should I?
No, well, how do you evaluate stand performance?
Yeah.
Well, I mean, we evaluate performance through tree improvement by selecting, but if you're collecting wild seed, you can expect, like from your seed zone, you can expect it's going to be very genetically diverse, because I know how our nursery system manages their seed, and they take it from private people and public from all over the state and merge it together. So assume that you've got a very good genetic base to work off of, and then the stand performance, then you need to do the silvicultural part. But if you've done that right, and you've managed your stock well, and you've gotten the right, you've got good stocking, good stock type that matches to the site, you should, any performance beyond that is up to you as a silviculturist to maintain. I think at that point you've covered your genetic basis. Does that make sense?
I think so. We'll see if we get another follow-up request. We do have two others. You mentioned that in the higher density Shelterwood style treatment there was greater genetic diversity because there were more mother trees. This next question is kind of an extension of that. Should we be using group selection instead of Shelterwood harvesting to preserve more genetic diversity?
You could, yeah. I think it would depend on, depending on the silvics of the species, if it prefers more, if that shade is beneficial or not. I think you would have to look at it adjusted to the silvics of the species, but you certainly could.
So if you get adequate light reaching the floor,
right.
With the group selection approach, maybe so.
Right, but like with eastern white pine, I think is Shelterwood. With red pine, more of a group selection if you were going to go that route.
Okay, are there any species or situations in Minnesota that would warrant special silvicultural treatment in regards to genetics?
Oh, regarding genetics, oh goodness. So, all right, there's the whole white pine thing with blister rust. That's a genetics question. Do we have the right material on the right site with enough genetic resistance to blister rust? I'm not sure. The other one I think about, silvicultural treatment and genetics. The other one that comes to mind is jack pine, gall rust out west, where gall rust can be out west relative to Brainerd area, not out west. There may be, to me that's more of a seed question though. Do you have the right seed on the site? Otherwise, if we were more interested in planting balsam fir can have some genetic issues that silviculture can address because it tends to be mid-story. The gene flow doesn't occur as easily in balsam fir, so if you were trying to propagate and improve the genetics of balsam fir, for example, you might need to do more opening up the stand to allow more gene flow in. But otherwise, and there may be some hardwoods that I'm missing, there's not a lot that I can think of right now, other than just getting the right seed source for the right site.
And what about elaborating a little bit on the blister rust question? I think there was some talk about that in our room here in Cloquet before you began. The Tree Improvement Cooperative, it sounds like is getting very close, or getting closer to having blister rust resistant strain. Is that correct?
We're making some very good strides. I've been analyzing some data from screening work that the Forest Service has been doing over at a Conto River Seed Orchard, and it looks like we have over 30 genotypes which have resistance that is as good or better than the Silver Splendor genotype that's being propagated for horticultural use. So the data is still being analyzed. I'm presenting it at a meeting in July, and we're going to try to have it peer-reviewed so that I can stand on the analysis with my peers. We are much, much closer than we've ever been, and I'm hoping that in a year or two we can start putting in some grafted orchards. And it's not immune. It will have improvements, so it's just traditional tree improvements, improved resistance to blister rust, but by no means immune.
All right, I'm not seeing any other questions. I will leave the chat open here for a couple of minutes, so if anyone has questions or if we have any questions from the room here, yeah, go ahead.
The literature you covered for selection is basically diameter limit stuff. Do what I consider a selection, where I would paint individual trees. I would assume that would change the genetics and stand more than just diameter limit.
Well, it depends if you're painting. So the question was, what if you're painting individual trees and you're doing that kind of selection? So the question would be, what's your criteria for selecting those trees? And that would depend. That would affect what the outcome is. So what kind of criteria would you be using?
Traditionally, you discriminate against poor form, forests, and stuff like that.
Okay.
Obviously, diseased trees.
Right.
I would think, in theory, in my head at least, you'd think that would move the genetics to stand faster. So then there might actually be more danger that you're actually going to, say I really don't like forked trees, right? So I mark all the forked trees.
Well, some of these.
Obviously, I'm simplifying things.
No, that's right. Forking trees and disease resistance. If you're selecting against trees with disease, you're probably, you may be benefiting the stand. But with forking, there's some characteristics that aren't genetic. So forking could have occurred from an ice storm that came through, and some trees maybe were taller and had their top cut off and forked. Others may not have. So you may be impacting the genetics, you might not be. You might be selecting against trees that had experienced some environmental experience that the others didn't. So you're not necessarily. I think a lot of those traits you're not going to impact it, and even if you were locally maybe changing the frequency of some of the alleles, there's probably a stand over 100 yards away that's going to send all this pollen in and recreate a lot of that genetic diversity. So locally you might be having an impact, but gene flow can undo a lot of that. And we have so many forests around that you may have some impact locally, but I doubt you're not going to have anything you need to worry about as far as the species goes. Anyone else?
All right, I don't see any other questions at this point. So again, I want to thank Carrie.